So in the previous lecture, we have learned about the basics of CRISPR, and in this lecture, we are going to learn about the practical aspects of CRISPR, how you can perform the experiments while you are in the laboratory. So, as you know that for doing the gene editing, we are using the CRISPR. Either we are doing the knock-in or the knockout. We are we can use the CRISPR along with other techniques. So what are the plasmids? If you want to, let's say, arrange a workshop, then you need to complete the process within a week or 10 days. You cannot organize a workshop for like 3 months. So when you are performing, when you are using the mammalian cells, what happened, because in my last lecture I told you that for doing CRISPR you have to do selection of clones in the 96 well plate. For example, if you have let's say 96 well plate, and in the last plate you will get single cell, and that single cell will form the multi colonies. If it is a mammalian cell, then it will take days or even a month's time, and that time you cannot spend in organizing a workshop. So it's a better strategy to use the bacterial cells instead of the mammalian cells if you are organizing a workshop. So how you can do that? For that you can just order the PRED, the plasmid, the PCAS9 plasmid from the AD gene. And what is this? And the catalog number of this plasmid is 42876. What is this plasmid? As you can see, this plasmid has a chloramphenicol resistance gene. So if the bacteria is harboring this plasmid, and if you are adding the chloramphenicol, the bacteria will grow. So this is the selection marker. Then these are the recombinant genes which help in the recombination. As I told in my last lecture, when you are doing the CRISPR, there will be either the non-homologous end joining or the homologous dependent recombination, in which you will provide the donor. So for that you need the recombination enzymes, and these enzymes are present in this vector and are under the control of the PBAD promoter. PBAD promoter is the arabinose inducible promoter, means once you add the arabinose, these promoters will activate and will help in the transcription of these REC genes, recombinase genes. Then this plasmid has the Cas9 gene under the control of the P2 promoter. These are the constitutive promoters, and the tracer RNA is under the control of P1 promoter. So I told in the last lecture that you need the tracer RNA as well, that is complementary to the palindromic repeats, and the Cas9 is present. So what you can do, actually you can just order this plasmid from the AD gene, but the only problem is this: if you are running a company, or if you are working in a company, AD gene is not providing their plasmids to private companies. So you have to be associated with the government organization like any university or institutes, so that you can order these plasmids directly from them. They are very cheap and you can just order it and keep it with you. This is the plasmid that contains the Cas9 gene. This I am telling you, if you want to organize a workshop and if you are facing a problem, then you can go ahead and do this. So what is this plasmid? One more time. It has the chloramphenicol resistance genes. It has the RAC genes which are under the control of the PBAD promoter. Why it is bad promoter? Because this is the arabinose inducible promoter. What happened is arabinose convert the sucrose. It inhibits the enzyme sucrase. So sucrose is not converting to glucose, and in the absence of glucose, bacteria may die. So that is why I think it is called the bad promoter, or but actually it is the arabinose inducible promoter. So your RAC genes are under the control of this promoter. So when you will add arabinose, this promoter will activate the transcription of these genes. Then Cas9 sequence genes that is under the control of promoter P2, and the tracer RNA that binds to the palindromic repeats that is under the control of P1 promoter. So this is the plasmid, just order it. And the catalog number, as I said, is 42876. Okay. Now, you need a plasmid also in which you can insert your guide RNA. Guide RNA means the RNA that will guide the Cas9 where it should cut the target gene. And this is against your gene of interest. And yeah, and your gene of interest should be near to the uh, protospacer adjacent motifs, which is PAM, or it should be near to the NGG, and we'll describe it. I've already described it in my previous lecture, but I'll describe it later as well. So for this plasmid, this is called the P-CRISPR SECP plasmid. Either you can directly purchase it from Dr. Grandi, who is in the University of Trento, Italy. He is working in University of Trento in Italy, and you can just email them and ask them if he can provide you this plasmid. He has this plasmid. Or else what you can do, you can purchase P-CRISPR plasmid and the PKM154 plasmid from the ad gene, and the catalogs number are like here. It has given 42875 is for P-CRISPR plasmid and 13036 is for PKM154 plasmid. So what is this plasmid actually? It has the kanamycin resistance genes. As I said, it is a selective marker. So if you have a bacteria and is that bacteria is harboring this plasmid, and if you are adding the kanamycin, because it is having the gene kanamycin resistance gene, so bacteria will grow if it is harboring this plasmid. So then it has the SECP gene. And what is this SECP gene? That encode the lever sucrase. This enzyme converts the sucrose to the toxic product. Okay, so if the bacteria, let's say if the bacteria is so let's say this is your what happened, let's say you have a bacteria cells, okay, and it is having this plasmid, which is, you can say, NP CRISPR sequi plasmid, and this plasmid, because it is having the kanamycin resistance gene, which is KNR. If you will grow this bacteria, this is, let's say, your agar plate, and you streak your bacteria and add the kanamycin here, then this bacteria is going to grow and you will get multiple colonies. Okay, this is the advantage of this. And the SECP gene, because it is having the SECP gene as well, it is having the SECP gene. Now if in this media you add the sucrose, and if this gene is present, then what will it do? It will convert sucrose to the glucose and the toxic product. Okay? And this toxic product is going to kill this bacteria. So if the bacteria is having this plasmid and you are adding sucrose, then this bacteria you will not get any kind of colony, you will get a plain plate. Okay, so this is all about so now, because when you will order this plasmid, it will be having the stuffer fragment here. Okay, it will be having the stuffer fragment. Let's say this is your stuffer fragment. means the fragment that can be removed with the desired sequence. Now you have to add gRNA, but when you will purchase, it will be having the stuffer fragment. Okay, and also you need to remember this plasmid has the restriction enzymes site for BSM B1. When you will order, you will get the entire knowledge about it from the vendor. So these sequences are under the control of P3, and as you can see, these are the palindromic repeats. This is the guide RNA. This is the stuffer fragment that you will replace it with the guide RNA. It is having the SECP gene, so if you are adding the sucrose, bacteria will die, and it is under the control of kanamycin resistance. So if you are adding the kanamycin, the bacteria will grow if it is having the kanamycin uh, in its medium. Okay, so bacteria is going to grow in the presence of kanamycin since it is having the kanamycin resistance genes. So what you can do, either you can directly purchase it from Dr. Grandi, or you can order these two plasmids separately, and based on some restriction enzymes digestion reaction you can ligate them and you will get something like that. Okay. Okay. So this is regarding this. Either you can order, you can just directly get it from Dr. Grandi, or you purchase these two plasmids and do some restriction enzyme digestion and the ligation reaction, and you will get something like that. But it is going to take some time, so it's better to get it from Dr. Grandi directly. So now, because as I said before, this is having the stuff of fragment, this is not having your guide RNA. So now you have to insert the guide RNA in this plasmid. And as I said, this plasmid is having the BSMV1 restriction sites. So you can simply, by doing the restriction digestion, you can incorporate your guide RNA in this plasmid. So now you have to make this p-CRISPR SECP plasmid to the p-CRISPR SECP gRNA plasmid. And how you can do that? So first of all, you have to identify a PAM sequence uh, near your target gene. So what you can do, if let's say if this is your sequence, let's say if this is your sequence, uh, it can be anything C N or CCT, you can say CAG. If this is the sequence you want to target, this sequence, when you are designing your guide RNA, this sequence should be near to the NGG. And your guide RNA, just remember, your guide RNA will not contain PAM sequence. Will not contain NGG or any other proteins based on the Cas9 source, from where your bacterial source, from where you are isolating the, from where you are getting the Cas9. So your guide RNA never should contain PAM site, but your target gene should be near to this. Your target gene should be near to this NGG sequence. Okay. Now, what you have to do now, we'll just go directly to this example. But this example I have taken from this website. So we are blasting the gene against the E. coli B21DE3 genome. So this is a site where you can just click it and you can get it. Or you can just go to the NCBI and type the accession number CP001509.3 and you will get the whole page that will be having a sequence. Here I am showing a portion of the sequence. When you will see the entire page, you can find this sequence on that page itself. So what we are going to do, here you can see CCN. It is the PAM, as I told in my previous lecture. It is not NGG, it is CCN. So what we have to do, we have to count the 30 nucleotides downstream of this sequence. If let's say if it is NGG, then we have to count the 20 nucleotides upstream of the sequence. But since this is CCC, which is the CCN sequence, so we have to count the 30 nucleotides downstream. So you have to remember, when there is NGG, Just count the sequence upstream when there is CC, and you need to count the base pairs uh, upstream from this sequence, and that is going to be your protospacer, because this is a protospacer adjacent motif, the motif that is adjacent to the protospacer. So this is protospacer, this whole sequence starting from C, and this triple C is your time sequence. So you just count it, and now as you can see here, C triple T triple 4A, and you can just match it, C triple T 4A, and all this is marked in the orange color, highlighted in orange color, and here you can see it is mentioned, it has mentioned in the red color. So you can see C triple T, and then it will go all the way around to GTC. Okay, so till here CAT GTC. Okay, now here you can see here, so this is like, let's say we count the C, C T T T A A A A. So what is going to be the complementary? It is like G A A A T T T T. And since this is 5 prime to 3 prime, this is going to be 3 prime to 5 prime. As I told you before, when you are ordering the primer, it should be always from 5 prime to 3 prime. So it should be ordered in the form of T T T T A A A G, and that you can see here, T T T T A A A G, like this. Okay, so this is going to be your forward primer. OMPF are the genes responsible for bacterial cells. So we are not worrying about these genes right now. We are just worrying how to do the experiment. So you have designed your reverse primer. So when there is a CCN sequence, that is going to be your reverse primer, not the forward primer. You have to be careful about it. And the next primer is going to be your forward primer. Okay. And because I told you this, in this plasmid, they have the BSM V1 sites. As I told in my previous lectures, you have to see this lecture if you want to understand why we are adding triple AC and triple 4A in the reverse primer. Because once you are having this plasmid, you will get something like this. You will get, when you will digest the plasmid, you will get something like this. Let's say, this is the, this is your sequence, and you will get in your forward primer. So you will get triple, no, you will get triple TG. Okay, and here you will get something like PTT. So there is an overhang. So in your forward primer, you have to add AAAC. In your reverse primer, you have to add AAA. Don't forget to add these sequences in your, so once you are done with this, you just add the 4AC in the forward primer and the 4A in the reverse primer. So but in case if you don't know, if you don't want to spend your time in designing your all these things, you can just simply order these primers and do the workshop. Okay, so now you have your primers. Now you have to anneal them, and annealing is very common. You can take the forward primer, reverse primer, 100 micromolar, 1 microliter. Take the ligase, take the P4 and polynucleotide kinase that will phosphorylate your sequence. Add the milky water, incubate it for 37 degrees. For annealing, add the 2.5 microliter or 1 molar NaCl and heat to 95 degrees for 5 minutes, and your annealing is done. So how much time you are going to take? 30 and 5, so 35 minutes. Okay, but you have to have all these things ready with you. Just make sure they are not expiring date. Now just allow them to cool to the room temperature by simply switching off the thermoblock, which is your PCR. You can also do in the hot bath, but it's better to do in the PCR machine. So once you have the 35 minutes time in between. In between this time, what you can do, you can just cleave it with the BSE1. I'm sorry about BSM1. This is the BSE1 restriction enzymes. Just purchases from the enemy, and take your plasmid, take this restriction enzyme, take the buffer and the water, incubate for 1 hour at 37 degrees centigrade. So what this enzyme is going to do, this enzyme will cut the this plasmid. Okay, which plasmid? This plasmid. This enzyme is going to cut this plasmid. Okay, then you have to dephosphorylate. Otherwise, what will happen? Even if you digest it, they will reannihilate because they are phosphorylated, right? So you have to dephosphorylate by the CIP enzyme, which is cough intestinal alkaline phosphatase. Do the reaction as per the manufacturer instructions. I have just told you. Just incubate this mixture, dephosphorylate, and for doing the dephosphorylation, you have to add the buffer, the enzyme, and your plasmid and the water. Okay. Okay. So now you have what you have right now. So just understand when you are digesting it, then when you are incubating it, then you just clean it and then do the dephosphorylation reaction. Okay. In the dephosphorylation reaction, you have to take this product along with the CIP and the buffer and the water. And what has it has been done? Now you have the plasmid which is cleaved. So how it will look like? It will look like something this kind of thing. It is cleaved. Okay. And since it is not phosphorylated, so they cannot reannihilate. Incubate for 2 hours. Just go follow the manufacturer instructions. Purify the plasmid by using the PCR cleanup system. You can purchase it on if you are working in the university, you might be having it. Then you have the cleaved plasmid. You have your ligated gRNA. What you have to do right now, gDNA, sorry. So what you have to do right now, take the this product, the cleaved pre-CRISPR sequi, which is the digested pre-CRISPR sequi. Take your phosphorylated double-stranded gDNA, add the ligase in the ligase buffer, incubate at 2 hours at room temperature, and then you are having the plasmid pre-CRISPR sequi gDNA plasmid ready with you. Okay, either you can proceed with the transformation, or you can just incubate these things for overnight at 16 degrees centigrade. Okay, the next day you can perform the experiment. Now you have to do the transformation, and for doing the transformation you need the bacteria, and this is the E. coli Th5 alpha, which is the chemically or electrocompetent cells that also you can purchase, or if you have bacteria you can make it chemically competent. And once you have it, you need to transform this bacteria first with this plasmid. Okay, which plasmid? This plasmid, or this plasmid, this plasmid, PKS9. And after you are done with this, you have to do the transformation with this p-CRISPR sequi plasmid, and this is again a very common form now of doing transformation. I have written the protocol, but you can just Google it and you will find the protocol of transformation. Okay, because this is the plasmid which is having the kanamycin, so you have to add in your LB agar the kanamycin. Can you incubate it? Definitely the colonies which are having the kanamycin are going to grow. You isolate the, you pick the colony, grow it overnight, and then do the manipulate. And then after you are having the plasmid, do not forget to do the DNA sequencing by using this primer. This is a very universal primer for this plasmid, so you can simply do the sequencing by using this primer. Okay. I have written here also the primer is universal and can be used for all CRISPR sequi gDNAs. Okay, you can just simply order it. And if you find your sequences there, you are ready to go with the mutagenesis steps. So you have right now plasmid, which is the p-CRISPR sec-pgDNA plasmid. You have the p-Cas9 plasmid, and now we can go ahead with the mutagenesis step. What you are going to do in the mutagenesis step? So you have to perform all these experiments in a serially also, in the respective fashion. You have to do this. First, you are going to order them. Then you are going to order these two primers, anneal them, digest this p-CRISPR sectoplasmid BSAI, okay, and then anneal it with this GDNA, which is ligated. And once you are done, you do the transformation just to be sure that everything is perfect. And then you are ready for doing your mutagenesis. Okay. So now what you can do here, I will go with the example here. We will see here. You can see this is the sequence we have already designed. Triple C, triple T, 4A. You can see here. It is triple C, which is your PAM site. This is triple T and the 4A. Okay. Triple T, 4A, double C. So here you can also see it is triple T, 4A, double C. So this is the sequence. So now because we are going to provide a donor DNA for inactivating the gene, so what you can do, you can just count the bases up till up here. Okay, you can count it A, C, C, C here till here. And what you are going to do right now, before this triple C, triple T, you need to delete, you know, this sequence. You are going to delete this sequence along with your what, TTA, as you can see, it is the stop codon. So once you are deleting this gene and adding this triple TA, you will get the, if it is once, this is the 30 base pair sequence, so total is 200 base pair. From here to here is, it is 200 base pair. So if there is an insertion of TTA, then this portion is going to be deleted. So 200 minus 30, you will get 170 base pair. Okay, but if there is no injection, then you will get only 200 base pair. So that's how you can differentiate between the clone that has been mutated and that has not been mutated. So here in this donor RNA, you have to delete this portion. This we do not need to take it. Okay. And we need to take just this sequence, and the sequences you can see here, A triple C, A triple C, and it will be CGAC. Double, where it is, CC, here I can see G triple A, CACC, GTP, ACCC, TT. So I think you have to take from here. A double C, GAC. T A G C C A G A C C G A C. So till here you have to include this portion. So that will be your 200 base pair. So from here till here, CGAC, double CGAC, it is 200 base pair. So instead of this sequence, you are going to add triple TA. Just remember that you have deleted the PAM site, which is CCN. This is the PAM site. You have deleted it. So the recleavage is not possible. So we have deleted the PAM site and we have added the stop codon to inactivate the gene. what you are going to do, you are going to order the primer A triple C and this, and the TTA total will be 200 base pair, and the reverse primer that you have to design the simply by doing the complementation. Following the complementation rule, as you can see here. Uh, So as you can see here, A is T, triple C is triple G, and T is A. So that you have to design, and there are softwares on AdGene by which you can do that also. So here, what you can see, you are providing the this RNA donor forward and the donor reverse RNA, and instead of the protospacer and the protospacer adjacent motif that you have deleted, you have added the stop codon, and that stop codon has no complementary sequence um, to the your target gene. So now you understand this. Okay. Design your mutagenic donor DNA in a way that will result in the deletion of 30 nucleotides within your target gene. So what we have done, we have deleted these 30 nucleotides and added the stop codon. Now we have removed the protospacer and the PAM region. So we have removed the triple C, which is the PAM region, and the protospacer adjacent motifs. Okay, write it here. So this is your PAM, and this is your protospacer, and we have removed everything, and we have what we have added here, we have just added the TTA, which is the stop codon. So in frame stop codon has to be inserted close to the 5' end of the target gene, and thereby it is inactivated. Note that the trinucleotide encoding the stop codon has no complementary sequence on the target strand. Okay, so that you have to be careful. So this is your donor DNA. The left part will bind here, the right part will bind here, and in between this is the TA, which is the stop codon. And similarly, this is the forward primer that will bind to the other strand of the DNA. What you are going to get? Now, once you have designed it, order it by the IDT or some other company, just anneal them, and that you can do by following, this many literatures on annealing of the oligos, or you can just simply take your double stranded DNA, anneal them, take the 10 microgram of each in a total volume of 20 microliter, 95 for 5 minutes, allowing the reaction mix to slowly cool down to room temperature by switching off the thermoblock, like the way we did it here, the ligation reaction. So you have to do the annealing reaction. You have to perform these things, okay, in the presence of ligase enzyme. So once you are done with this, now you are ready with your experiment. Okay, you have the PCAS red plasmid, and for performing the mutagenesis, you have the pre-CRISPR SecPG DNA, you have the donor DNA. Just do the colony PCR, take the forward primer and the reverse primer, take the tech polymerase, do the colony PCR, pick the individual colonies, transfer directly to the PCR reaction mix, and the PCR reaction mix will be having everything, the MgCl2, the tech buffer, and then follow the denaturation, 30 cycles of denaturation and annealing elongation, and then the final elongation. And then you verify the mutants by loading the 4 to 8 microliter of each PCR on the 2% agarose gel in the run electrophoresis. So what you are going to get here, so if there is no deletion of this protospacer in the PAM, then you will get a 200 base pair. If there is a deletion and you have inactivated your target gene by incorporating this stop codon, you will get the 170 base pair product. So that's how you can differentiate it. Okay. So here you can see this is a wild type genome, this is knockout mutant. This experiment you can very well complete in 7 days, I believe. Okay. So now, once you are done with all this, and if you want to cure your plasmid, your um, Curing of the plasmid. What you can do, just pick a mutated colony and incubate it with the the kanamycin, because this plasmid is having the kanamycin resistance gene. And also because this is having the SECB, so you just incubate this mutated colony in the presence of sucrose and the chloramphenicol. And what it will do, incubate, then do the vigorous shock shaking overnight. You will get a culture. You can prepare a glycerol stock, and then you can just um, control loss of the p-CRISPR SECP DNA plasmid by adding 10 to 20 microliter drops of the 2 LVAR place, one containing the chloramphenicol and the one containing both. So no growth should be visible on the plate with the chloramphenicol and the kanamycin recombination, indicating that all bacteria had lost the p-crispy or SECP gDNA plasmid. First of all, when you have the mutated colony, you are adding the sucrose and the chloramphenicol and the kanamycin. There you will not get any kind of growth, because a SECP gene is present. So SECP will convert sucrose to the toxic product. And again, the SECP plasmid, I think it's not chloramphenicol, it's a kanamycin. Let me just see this. This is a kanamycin. So you have to grow these cells in the presence of kanamycin. You will not get any kind of growth if you will, because it is having the kanamycin resistance gene. And if you are adding the chloramphenicol, then what happened, the sucrose will be converted into the toxic product, and since the plasmid is not having any resistance genes for the chloramphenicol, they will not grow. And in this way you can get a colony that is not having any p-crispy or secretary. Okay, if you want to cure it. So it's called the curing of this plasmid. Curing of the p-cassette plasmid, because this plasmid is having a gene for the chloramphenicol. So just add the kanamycin, and this you will not get any kind of growth. So this plasmid is having a chloramphenicol resistance gene. So if you will add the kanamycin, it will not grow. So you just add the containing the kanamycin, and no growth should be visible when you will add the, not the chloramphenicol, but the kanamycin, because it is having the chloramphenicol. I have written it wrong, just mention it, cross it. This is like kanamycin. So that all bacteria have lost the p-cassette plasmid. So that's how you can perform the workshop in your lab. You have to, what you have to do actually, just order these two plasmids from AdGene or directly get it from Dr. Grandy. Do some order these two primers. Order these two primers and just follow the equations, and you will get everything in pretty much in a perfect way. And then you have to order these two plasmids as well. Okay, here you can see in the donor I have added the TTA and deleted the protospacer and the protospacer adjacent motifs. Do the ligation reaction, run the echo, and you are done, you are good to go. This is regarding the practical aspect of CRISPR, and this lecture I am giving because I have seen many scientists want to organize a workshop, and then they are not aware of how to organize it. So this lecture is for the those people who want to organize a workshop and who want to spread their knowledge to the coming generation. So thank you very much for this, and in my next lecture we are going to discuss about the 3D cell culture. Thank you very much.